So we did the transformation of PCM 189 Bax yeast. My name is Logan.
I'm Isaiah.
My name's Gabby. And my name's Gabby.
Next slide. So we used yeast because it grows quickly. It takes about 90 minutes to divide and duplicate. We also used it because it's pretty easy to propagate. Yeast is also one of the eukaryotic organisms that's had its genome completely sequenced, so a lot is currently known about it. Most yeast have shuttle vectors that can carry DNA of interest. These shuttle vectors are usually plasmids that are capable of propagating in 2 different species, such as E. coli and yeast. So on to our proteins/genes of interest. The B-cell lymphoma family, better known as the BCL family of proteins. This family is made up of proteins that have both anti-apoptotic and pro-apoptotic traits. The first gene of interest is BCL2, which inhibits apoptosis by inactivating Bax and BAC proteins, which are the pro-apoptotic proteins that help release the cytochrome C from the mitochondria that causes the cell death. Specifically, we'll be looking at Bax, the BCL-associated X protein. It's a member of the BCL2 family that forms heterodimers with BCL2 and functions as an apoptotic activator. Bax is activated and oligomerized at the mitochondrial outer membrane to help mediate its permeabilization, which is an essential step in apoptosis. Bax forms oligomeric pores in mitochondria's outer membrane to help release the cytochrome C. This protein also promotes apoptosis. Its function is to oppose the BCL2 gene specifically. The expression of this gene is regulated by the tumor suppressor p53 and has been shown to be involved in p53-mediated apoptosis. So why are we interested in this particular family of proteins so much? The BCL2 proteins are regulators of apoptosis, so they regulate and mediate the process of the mitochondria contributing to a cell's death. This is known as intrinsic apoptosis pathway and is required for normal embryonic development and the prevention of cancer. If apoptosis is interrupted, the cells wouldn't die, they would continue to grow and eventually cause tumors, leading up to cancer. Next slide. Our objective of this experiment was to transform the yeast with the PCM 189 Bax plasmid and to verify our successful transformation via methods that Gabby will be discussing shortly.
Okay, so one of the first steps in our experiment was to transform E. coli bacteria with our PCM 189 Bax plasmid using horizontal gene transfer methods, followed by amplification in a selective medium and purification of the plasmid. Then we analyzed our purified plasmid using restriction enzymes to confirm that we indeed obtained the correct plasmid so we can effectively continue with our experiment. After analysis, we finally transformed yeast with our PCM 189 Bax plasmid using similar method to those used in the transformation of E. coli. Okay, next slide.
Thank you, Gabby. Looking at the results of our experiment, our initial transformation of the PCM 189 Bax plasmid into E. coli yielded positive results. The growth of the bacteria in ampicillin confirms the successful transformation of the plasmid due to its ampicillin resistance. This also ensures the plasmid is valid and usable for our yeast transformation moving forward. As you can see in this next slide, the growth of yeast in doxycycline and in the absence of uracil indicates a successful transformation of PCM 189 Bax into yeast. This is further confirmed through restriction analysis of our agarose gel electrophoresis. As you can see, when cut at the BAM-HI and X-Hall sites, we recovered the correct bands in our gel electrophoresis. At about 30 colonies, our growth was relatively limited when compared to some of our classmates' growth of PCM 184. But this is expected due to the apoptotic nature of Bax. The transformation of PCM 189 Bax into yeast allows for the future analysis of the plasmid's function in eukaryotic cells. It also allows for better understanding of Bax and cell apoptosis, as well as the relationship between Bax, the pro-cell death proteins, and BCL2, BCLXL, the anti-cell death proteins. It also allows for insights on how to prevent tumor creation and progression in mammalian cells. I'd like to take a moment to acknowledge and thank Dr. DeJean and the Fresno State Chemistry Department for allowing us to partake in this lab. Thank you.